This video is the second in a series of 3 that provide an overview of dbGaP's submission process. The other videos are Part 1, Register Your Study, and Part 3, Review and Release Your Study. If you want more background on dbGaP, see the recent video, What is dbGaP? A link to that and the other videos in this series is at the end of this video. In the first video, Register Your Study, we ended with your GPA, the Genomic Program Administrator, initiating registration of your submission. Once that is complete, the designated PI, or Principal Investigator, and the PI Assistant will receive email invitations to access the submission portal. They need to accept this invitation within 7 days, even if they are not planning to submit for some time. Once inside the submission portal, the first step is to complete the study data outline, where the submitter will indicate the expected data components to be submitted for a study version. The major data components, which we'll refer to throughout the video, are phenotype, study documents, high-throughput sequences, molecular data, and association analyses, which are aggregated summary results from genome-wide association studies, or GWAS. Once the study data outline is completed, the study will be assigned a PHS study accession, which is a permanent identifier that can be used in publications. Please make use of our submission guide, templates, and pre-validation tools such as GAP tools, because cleaner data means faster processing and release of your study. Let's see how the data are organized by briefly reviewing dbGaP's data model, beginning with the phenotype component. All of these blue rectangular tables are part of the phenotype component. There is a row for each subject or sample, and any number of columns with the phenotypic variables. And every dataset must have a corresponding data dictionary that minimally provides descriptions of the variables, units of measurement, and code meanings for coded values. Within the phenotype component are 2 core files, subject consent and subject sample mapping. Broadly, these files are used to assert the subject and sample identifiers and the consents across all data components with individual level data. More specifically, the subject consent dataset is a complete list of study participants for which individual level data will be available through dbGaP authorized access. Each person should be listed with one unique, de-identified subject ID, a single consent value based on the data use limitation, and the person's biological sex. The list of subject IDs should also include publicly available genotyping controls, such as HAP maps, and linking family members having no individual level data, but which are required to complete the pedigree. These subjects are the only ones that can be assigned consent equals 0. All other subjects must be assigned a consent value using an integer greater than 0. A pedigree file should be included if there are related individuals in the data. The subject IDs in the pedigree should include all related subjects and their linking family members. This helps elucidate the mode of inheritance and aids in genetic variant discovery. If you would like to link subject IDs across multiple dbGaP studies, there is also an option to use aliases. This brings us to the other core file, the Subject Sample Mapping, or SSM dataset. This 2-column table shows that each sample belongs to a consented individual. The SSM maps subject IDs with their corresponding de-identified sample IDs, and a person can have multiple samples. Ideally, a sample ID is for the final aliquot used to produce the molecular and high-throughput sequencing data. Each sample should be further described in the Sample Attributes dataset, with minimally the variables such as body site, analyte type, and tumor status. Any number of variables may be added, and there can be more than one Sample Attributes file. We also expect subject phenotype data to be submitted. These are sometimes referred to as metadata. These data help with the interpretation of the submitted multiomics data and include clinical information, exposure data, and descriptive information, for example, the protocols or methodologies used. They can also have any number of variables and any number of files. That's all for the typical phenotype component. For other variations, please see the dbGaP submission guide. Be aware that for each study you submit, you will be working with a dbGaP phenotype curator. And if you have molecular data, you may also be working with a dbGaP genotype curator. Let's look at a second component, study documents. Please submit documents such as protocols, questionnaires, and consents if they can be displayed publicly without copyright issues. They will be marked up in XML and linked to relevant phenotype datasets on the public web page so that others can more thoroughly understand your study. Now let's look at two more components, high-throughput sequences and molecular data. If submitting high-throughput sequence, here are some considerations. Overall, the process will take 2 steps. Step 1 is to submit a valid sequence metadata file that is used to define the sequence files that will be submitted. Once those are loaded, step 2 is to submit the sequences. Here are some examples of high-throughput sequence: whole genome, exome, and targeted sequence, and RNA-seq, ATAC-seq, and methylation-seq. If RNA sequences are submitted and expression counts are available, please determine if the expression data can be submitted to NCBI Geo, the Gene Expression Omnibus, where the data will be publicly available. If not submitted to Geo, the RNA-seq data goes to dbGaP behind controlled access, and the expression counts are submitted to the molecular data component. Similarly, if whole genome, exome, or targeted sequences are submitted, please also consider submitting VCFs, or variant calls, which are more commonly downloaded and used. Please upload VCFs under the molecular data component as well. Even if no sequences are submitted, expression counts and VCFs can still be submitted under the molecular data component, along with SNP arrays, imputations, and various omics data, such as proteomics, metabolomics, and methylomics. Keep in mind that every sample ID listed in the molecular and sequence data must map back to a consented subject in the subject sample mapping dataset. If the sample IDs are not in the files, then please submit a 2-column table with the sample IDs mapped to the file names. And if you have genomic summary results from genomic association studies, those could be submitted under the association analyses component. These include linkage and burden testing on genotypic and phenotypic traits. Since these are summary level data, not individual level data, there are no subject consents tied to these analyses. So during the study registration process, the study should be marked as either Genomic Summary Results, or GSR, sensitive and only distributed through authorized access, or marked as unrestricted and available through dbGaP's public FTP. That's all for Part 2. For more information, please see the dbGaP submission guide, and you can send questions to dbGaP-sp-help@ncbi.nlm.nih.gov.